In this video, we'll be having a look at the structure of a plasma membrane. Now, plasma membranes form that barrier between the inside of a cell and the outside. And what this does, it helps a cell to regulate its internal environment, which could be quite different from the environment outside the cell. And a plasma membrane forms that effective barrier between the extracellular fluid and the cytosol. And really the key feature, the key molecule that we have, or a group of molecules, form the phospholipid bilayer. So this gives you some clue, the name phospholipid, right? And phospholipids are interesting because they have two parts, and this is the phospholipid right here. And it has a polar or a hydrophilic head. So the hydrophilic head likes to be near water molecules. And then we have these two tails, which are the nonpolar or the hydrophobic tails, which tells us it does not like to be near water. So it has to interact with water in both the fluid components, and it has to do this without falling apart. And it has to also have the ability to repel water, keeping the extracellular fluid on the outside and the intracellular fluid, or the cytosol, on the inside, and keep the two separated. So we have this sort of very unique situation that, here we have a plasma membrane. And on the inside, we basically have a water environment, the cytosol. And on the outside, we have a water environment. Now, of course, suspended in the water could be quite different things. And one of the jobs of the plasma membrane is to make sure that we carefully regulate this water. Because if you imagine, if we allowed water to rush out of a cell, and if too much water rushed out of the cell, that cell would basically shrivel. If too much water rushed into a cell, the cell would swell up and potentially explode. So one of the big features is how do I put together a membrane that is tough enough to survive, has some flexibility, and yet can separate the two water layers. And this is the perfect design. As we'll see, we have as I said, a polar and a non-polar region of one molecule. So when we look at phospholipids, they're amphiphilic. The phosphate group, so this is the phosphate group, it faces the water zones on the outside and the inside. And then there's two fatty acids that come together, and these are the hydrophilic bits. So if we were just to take a group of individual phospholipids and throw them in water, what would naturally happen is the water parts would hang out with the water, and the non-polar hydrophobic bits would be repelled by the water. And you can see this if you have phospholipids and you throw them in water like they did here in the test tube. You can see what happens, that all the molecules, well, the water-loving bit will hang out with the water, and the water-hating bit, hydrophobic bits, will move away. So you can see how I don't need any special energy, any special mechanism, other than just the basic chemistry of this molecule to ensure that I can have the phospholipids hanging out near the water, because they want to, and the phosphates, I should say, the phosphates hanging out near the water, and these lipids hanging out away from the water. No, so that is one layer. But as you can imagine, these things can also self-assemble in a different way. That they can self-assemble that these water-hating, hydrophobic bits will all face towards each other naturally, because they want to be away from the water molecules. And so in many ways, a cell membrane is sort of designed to almost self-assemble to some degree. because the phospholipids will naturally line up that the water-loving bit will be near the water, and the water-hating bits will be away from there. And how do they escape the water? They basically form their own little environment by this phospholipid molecule lines up with another one, and they basically have the ability to exclude water within there. And we form a bilayer. Why a bilayer? Bi means two, so there we go. A two-layer phospholipid membrane with the non-polar tails facing each other and the polar components facing the water. All right, so that's one of the big components of a cell membrane, but it's not the only. Because remember, these are phospholipids, so we're looking at a fatty type material. We have to make sure that cell membranes are kind of in that perfect zone of not being too liquidy and not being too solid. And we also have to have the ability to move stuff out of the cell and in the cell. So here's something called the fluid mosaic model of the plasma membrane. And the fluid, there's a lot of clues in here. Fluid, okay, mosaic, it's made of parts of the plasma membrane. The fluid mosaic model really indicates that we're looking at a dynamic fluid structure. Now here's one of the problems. We're looking at this and you see all these individual phospholipid molecules. It makes it look like they're solid, that this whole membrane is almost like the outside of a chicken egg. It's not. This is a fairly fluid cell membrane, and that's wild to think that your cell membranes are somewhat fluid. That certainly, some of the things that are incorporated within here almost float around as if they're icebergs in an ocean. So there's a lot of potential movement within here. So this thing's called the fluid mosaic model. Now, we can already start to see from this diagram as I'm looking at a cell membrane that certainly the phospholipids make up the much of the cell surface. But we can start to see there's a lot of things that are either on the outside or some things that are on the inside of the cell membrane, or some things that pass through the cell membrane. And also some of the things that are embedded in the cell membrane have other types of things attached onto them. And we can have a look at that. Is that, we see that we have a lot of proteins that are embedded in a cell membrane. Now, just to do a general categorization, the proteins that are only hanging out at either the outside or the inside of the cell, they're peripheral to the cell membrane. We call peripheral proteins. That's a peripheral protein, that's a peripheral one because it doesn't go all the way through. We also have the ones that are largely embedded within the cell membrane, integral proteins. And then we have another group, which are the transmembrane proteins. Trans means they go definitely from inside the cell all the way through the cell membrane, and they're on the outside. We can also see that attached onto a lot of these proteins, we can have other substances. And one of the most common things are carbohydrate chains that are attached. And by the way, this is sort of shorthand for a sugar molecule, right? So that's a carbohydrate chain that we see right there. Now, sometimes some of the phospholipids actually have a glycolipid instead. So you see this? This is a chain of sugar molecules, okay? And it's actually attached to the fatty acids of a phospholipid. So here's another type of structure we can see right here. It's a glycolipid. And then if I look carefully, you can see embedded in here this structure, this multi-ring structure. And certainly if you've ever done biochemistry, you would automatically recognize this as a very unique shape, and we'll kind of get used to seeing this. This is a cholesterol molecule. And you're going, wow, cholesterol. We tend to think, cholesterol has a very sort of negative thing in popular press. It's, about foods without cholesterol, things like that. But actually, cholesterol is a very interesting molecule. A lot of hormones are actually derived from cholesterol. And here's another important bit. You can see there's a cholesterol molecule there, cholesterol molecule there. Cholesterol plays a very big role in stabilizing cell membranes. Now it's like it's like many things in life, that a little bit's not good, too much is too much, and that middle zone is just right. So we need a certain amount of cholesterol. We don't want too much in the cell membranes to overly stabilize it, but we need X amount. And what the cholesterol does is, remember, this is called the fluid mosaic model. So these phospholipids tend to be almost watery and kind of want to move around, and things may float around through them. And that's okay, but we want to make sure it doesn't get too liquidy, the membrane, because then it's very prone to rupturing. So cholesterol plays a big role in helping to stabilize that. So, as we said, there's a number of membrane proteins, and there's integral, and they span the entire membrane. Sometimes the name integral and transmembrane are often used synonymous. And peripheral are only found on one side of the membrane, so either on the inside or the outside of the membrane. Now, there is a group of membrane proteins, and we can start looking at some of the functions of these. Channels, very important. These are transmembrane proteins that allow certain substances to cross a membrane and pass in or out of the cell. So you can see these are very important for materials get in a cell, get out of a cell, or it also, some of these help regulate how much of stuff is in a cell or outside of a cell also. Now, so we were looking at channels right there, and channels are and I should just spend a second here. When they're open, they allow certain things based on their chemistry or size, I should say, to pass in or out. We also have these things called carrier proteins. And what they do is they're an integral protein, so they are embedded in the cell membrane, and they bind. Now, they don't bind to everything. They're designed to bind to certain things, but when that certain thing binds onto it, they change shape, and that allows them to directly transport substances in or out of a cell. So you can see here that the carrier protein is in the open form, and a substance, not any substance, but the one that it's designed to carry, binds. And when it binds, then the protein's going to change shape, and you notice what it does is allow the substance to go inside. Now, you notice that it doesn't allow things to freely go in. So in order to get in, you actually have to bind, get the shape change, and then enter. So it helps you regulate what's coming and going from there. Now, when I'm looking at these, there's another group called receptors. And receptors bind to chemical messengers. Ligands, okay? To ligate means to tie something, a ligature, okay? It's one way to help remember what it is. And in this case, the receptors, if they're a chemical type of receptor, they require a ligand, a specific ligand, not just anything will do it. And it triggers a sequence of events within a cell. And so here's just a basic example. A ligand binds on, and that may be a signal, because you can see that the channel's closed, but if a ligand binds, it opens the channel and things can cross in and out. And then the other big group that we have are enzymes. And we already know enzymes speed up chemical reactions. And remember, enzymes are specific for a substrate. So there are a lot of enzymes that are embedded in many cell membranes. And the substrate comes in, binds, and then the enzyme, through its acting as a catalyst for the chemical reaction, the product is then produced. And then we also have structural support. Okay. Now, remember earlier on I mentioned that there's a thing like the cytoskeleton that runs through the cell. This is a place where things like the cytoskeleton can attach on. Okay. So these membranes that are embedded into these proteins that are embedded into the cell membrane, by attaching different things to them, can help to stabilize the cell membrane or cause it to change shape. So structural support is another big function of membrane proteins. It helps to give cells their shape, helps to maintain structural integrity. And there's still more. You can see that membranes play a huge role in cells. Sometimes what they do is that they help to anchor cells, because most cells don't get up and walk around the body. They're sort of stay within the tissue or their tissue. So they can link adjacent cells to one another, and they can anchor a cell within a tissue, or they can anchor it to another cell, and sometimes these actually allow for cell-to-cell communication. Now, we mentioned a little bit earlier, but when we look at other membrane components, that we often see lipids, carbohydrates, glycolipids, glycoproteins. So, as we mentioned earlier, things like cholesterol, which is a lipid, it helps to stabilize the membrane and keep it just fluid enough without falling apart. And especially during temperature changes, this plays a vital role. We also have glycolipids and glycoproteins. Now, glyco gives you a clue. Glycogen, right? Carbohydrates. These are carbohydrates. So not surprisingly, when I attach a carbohydrate to a lipid, it takes on the name a glycolipid. A carbohydrate onto a protein, I get a glycoprotein. And these play a number of roles. Some of the roles they play would be things such as cell recognition and have roles in the immune system. Okay, I'm not going to spend much time here, but if you have a look at the book, they often sort of help you understand this within the context. And not surprising, there are many drugs that have a role in attaching onto cell membranes, because cell membranes are how a cell interacts with the outside world to it. And also, it's at a cell membrane that many chemical signals come to tell a cell what to do. Should I turn on a certain pathway, etc. And not surprisingly, many drugs are designed to look like receptor ligands. Remember what a ligand is? That many receptors need something to bind onto it, which we call a ligand. And often you'll hear in the world of pharmacology, agonists and antagonists. So if you're a drug that's being designed to bind onto a specific ligand in a certain group of cells, an agonist mimics. So it pretends it's that ligand, right? So it mimics the ligand's actions. And in that case, it stimulates a receptor. And some examples of this would be narcotic painkillers, such as morphine. Because how morphine works, and the opioids, is that they actually bind on to receptors that are in our cells, because we naturally produce something called endorphins. So morphine pretends it's an endorphin, has the ability and the shape to attach onto a ligand and stimulate the receptor due to its work. Antagonists, okay, they inhibit a a ligand's action by blocking the receptor. So if I come along and block that receptor with a chemical, then the receptor cannot hear its normal signal and it will not work. So this is the way really typically most antihistamines work, is that they come and they bind the histamine receptor. So the signal, which is actually histamine itself, can't bind onto the receptor and cause your runny eyes and your wheeziness and mucus production in your nose, etc., etc. So these are two really important terms that you'll hear in pharmacology all the time. Agonists, they mimic the action. Antagonists inhibit the action. Okay, we're going to leave it there and we're going to move into transport across cell membranes in the next video.